Good evening, everyone who has joined us today. Welcome into the first of our June seminars for the Greater Houston section of the American Chemical Society. My name is Dawn Friesen. I'm an alternate counselor for the section and am very pleased to host this seminar on behalf of our chair, Sophia Mohammed. With that, I am very happy to introduce our speaker today, Dr. Raúl Hernández Sánchez from Rice University. As a little bit of a bio, he was born in Chihuahua, Mexico, and did his undergraduate working through Caltech, Southern University of Denmark, and received his undergraduate from ITESM campus Monterey. At that point, he moved to Cambridge, Massachusetts to pursue a PhD in chemistry at Harvard under the mentorship of Professor Ted Bentley, and then started a Columbia Nano Initiative postdoctoral fellowship with Professor Colin Nuckolls. Prior to joining Rice, he was an assistant professor in the Department of Chemistry at the University of Pittsburgh and joined the chemistry faculty at Rice in the summer of 2022, where he is the Norman Hackerman Welch Young Investigator Junior Chair. At Rice University, his group's research interests lie at the interface between synthetic organic and inorganic chemistry to create novel functional materials and catalysts capable of activating small molecules at polynuclear reaction sites, the creation of novel contorted aromatics, and the design of anion receptors for the removal of toxic chemicals from our environment. Most notably, Dr. Hernández Sánchez was named one of Chemical and Engineering News Talented 12 for the class of 2023, and we are very pleased to have him speak to us tonight. With that, I'll pass it over to you.
Well, that was a fantastic introduction. Thank you very much, Don. And thanks to those that are connected right now synchronously, and then those hello to those that will look at this later on. So I usually like to start by acknowledging the people that do the work. I'm the guy that presents it and has a lot of joy at looking at the data and analyzing it with them. But shown here are the people, the students in my group that really make all of the beautiful research that I'm gonna show you in tonight's seminar. So with that, let me tell you that our group is centered around three different topics: synthesis, organometallic chemistry, and catalysis to design novel materials. The idea ultimately is to be rooted in the synthesis of materials so that we can impact novel organometallic species or catalytic systems that will eventually have some purpose. This slide essentially shows you the three research areas that we work on. On the left, you have a topic that we call tubularines. And that's essentially centered around the idea of creating contorted aromatic nanotubes that we can synthesize in a well-defined fashion. We use the same scaffold that we're using for the tubes. We're using it to create anion receptors. Here shown in this crystal structure is methyl sulfonate. Let me clean this up a little bit. So that's methyl sulfonate hosted within this cavity. And I'm going to tell you towards the end why we're interested in anion receptors and what exactly we're using them for. And then in the middle of my talk, I'm going to be telling you about cluster compounds and how we're engineering these type of compounds where you have metal-metal interactions that drive, hopefully, small molecule activation. So let me begin with the idea of bending aromatic systems. And so in that regard, you have one of the most famous materials known to man, and that is carbon nanotubes. Carbon nanotubes, since their discovery, have been used in composite materials. Here I have just two examples. The second one somewhat exotic, but carbon nanotubes have been used in paint to essentially use them as anti-fouling agents. They have been used in spacecraft as in ESD shields, and in many other applications. Now, the real problem here is in the synthesis in how to design tubes that have a specific length, a specific diameter, and third, a specific chirality, right? How do we especially engineer nanotubes with a well-defined property? And so if we look at two of the common geometries that you find in carbon nanotubes, you have what is called the armchair and the zigzag nanotubes. And since many years ago, these small fragments of a nanotube or benzenoid fragments of a nanotube have been targeted or having the target of synthesis. The one on the left is called a cycloparaffenylene, which was synthesized for the first time in 2008. And then the one on the right, which is a cyclazine, or now more commonly known as belt arene, proposed since the mid-50s and just recently reported in the early 2021. So the Chem Archive papers came in December 2020, but then the actual papers came right at the turn of the year. Anyhow, so the basic question is, how do you synthesize contorted aromatic systems like this, that you can eventually go back and then create tubes that look like that? So in my group, the approach that we took was to use a template shown here. You have a resorcinarene that we're derivatizing with this dichloroquinoxylene to form this compound that we call colloquially octobromo. This octobromo species, under Suzuki conditions, yields the contorted aromatic species shown here. We were able to crystallize it. That's the molecular crystal structure of that compound. DFT calculations tell us that it has about 90 kcals per mole of strain energy. Now, to put that in perspective, a CH bond in methane has a bond dissociation energy of 104 kcals per mole, right? That is the bond dissociation energy. Here we have a compound with 90 kcals per mole of just strain, mechanical strain, by distorting the aromatic system. Now, we investigated this species and realized that you can reduce it 4 times or up to 4 times. It has an interior cavity shown there of about slightly longer than a nanometer. And when we were getting this data, we were thinking, what can we use them for? So one of the things we envisioned was, well, these are nitrogen atoms shown right here. And if you hypothetically have acid around, you can potentially protonate this. And if you are lucky enough, you can potentially protonate it twice in an adjacent fashion to form these kind of species. And if you can reduce it, as we proved the compound by itself, then you can potentially evolve H2, right? So that was the hypothesis. And so we started thinking and collaborating with people in the field, in this case specifically with Professor Echegoyen at UTEP. And what Olivia found was that this compound has a very low onset potential for hydrogen evolution. It's actually comparable or even better than the best fullerene species that they had studied at the time. That is a scandium nitride D3C68, which is something that is extremely difficult to obtain. And what we realized is that from computational studies done by my colleague, Professor Peng Liu at the University of Pittsburgh, was that one of the key features here that allowed us to really get catalysis going was the fact that you can protonate the contorted aromatic system, liberate some of that strain energy, and then from there, the reaction then occurs downhill, right? And at the time, and we haven't actually published this, but this has been data from now almost 2-3 years old. There was hints in the literature that you could do this with CPPs. However, asking some of the leading scientists in CPP chemistry, they told us that one of the things that happen is you see a 1-2 shift in the aromatic system, and the system is done, right? All of that strain energy, as opposed to funnel it into making a hydrogen bond by addition of electrons, you actually funnel it into a 1-2 shift, and there's no more strain. Not here, because it's aided by essentially this scaffold that keeps the structure intact, or the structure in that configuration. Cool. So we think of this as a new design tool for HER, and we're still exploring some of these features with other compounds that we're making as we speak. Now, sometime after we finalized the synthesis of this compound, which I'm calling P4 for 4 para-connected phenylines, we started thinking, well, what if we prepare on purpose the old meta version, right? As shown here. My student Saber and Victor at the time figured out the conditions to make that happen, which were really not that different from the conditions to make P4, other than just changing the starting material. They were able to obtain the crystal structure of that compound, so M4, and also all of the intermediates in between those two bookends, the P4 and the M4. All of those isomers are shown here. They're crystal structures, except for one that we have a lot of troubles to get. But for the, for what it matters, the NMR is pretty indicative. Anyhow, so the important aspect of doing this chemistry was to really try to understand what happens with the electronic properties when we're going from this all meta-connected tube to this all para-connected species. And so the answer to that question comes really handy when looking at this picture that my students sent me on a Saturday afternoon right after lunch. And I was super happy and stoked to see it because, well, number one, this is the type of images that are in the literature for when people were synthesizing quantum dots, right? This is in the late 80s, 90s. However, here we have a molecular system, right? So all of these are molecules that only differ in the connectivity, as shown right here. And so the way we understand this, and I'm not going to go into all of the DFT studies that we've done, but the way we understand this is that you're essentially limiting, as you go from right to left, you're limiting the size of the conjugation pathway, and therefore changing the box. So you can think of, the particle in the box example. And so you're shrinking the box more and more, and therefore you have this blue shifting of the emission as you go from the fully conjugated species to this metaphor, which now you have a large dihedral angle or torsion angle right there. Anyhow, so we're in the process of finishing this up, but at the same time, we have made some changes to the synthetic protocols that we're exploring. So I told you that everything we're doing starts really from a scaffold that looks very much like this one. So it's a raceminerine. Now, doing a small changes in the particular synthetic protocol, so changes right here in this connectivity, we were able to obtain these compounds that are somewhat like a net, right? Difference is that the strain is now minimal, and now we can get yields that are in excess of 70%. Versus before, we were having yields on the tubes that I just showed you at the order of 1 to 2%. Now, why is this relevant? Well, we figure out that this net is particularly a good host for fullerenes. And this is before I came to Rice. This is when I was in Pittsburgh, right? So, because people ask me, do you get paid more just because you're working with fullerenes at Rice? Not really, not really. I mean, I wish. Anyhow, the point is that there's really strong binding of fullerenes by these particular nets. So here you have the free net, zero equivalence of C60 added. This is the acetal resonance. So it's one of the resonances pointing right here towards the C60 molecule. And as you can see, as you add equivalence of C60, you start seeing the coexistence of the host gas data and the free host, meaning that there's essentially in this solvent no equilibrium. The moment you add C60, the moment the equilibrium is pushed all the way to the right, you form the host gas complex and you have free host. There's no back and forth. That's why you don't see the monotonic change that usually you observe in a titration experiment. Well, not here. And in fact, based on these experiments and done via NMR, we know that the association constant is in excess of 10 to the 6 per molar. So when we got these results, we were thinking, how can we take advantage of these numbers, right? So we started thinking, well, carbon soot. I mean, carbon soot is essentially just partially burned carbon, right? The leftover. You have you're saying that in that picture, right? And so we thought, what if we mix this net in a solvent plus carbon soot? Will we be able to extract fullerenes from carbon soot, which in other ways, another way to say it is from garbage, right? And so this is the modeling that you obtain, right? This modeling goes from 2000 to 9000, and essentially you only see a few peaks in this modeling, right? And if we center in this region of the spectra, we can see that this particular net is essentially sequestering all of the different fullerenes from C60 all the way up to C96 in one go, right? This is pretty remarkable because you have one host being selective towards fullerenes, because you can imagine, right? This is carbon that has everything that you can imagine under the roof, right? It has aromatics of small size, large size, medium size, everything, right? But instead of grabbing all of those, it's actually just grabbing only aromatics that have that, spherical or spheroidal shape. One of the most prominent ones, which are these ones, are shown here on the top for C60, monomer, dimer, and C70. Anyhow, so in the future, what we want to do with this is to start designing membranes that can sequester this selectively. Anyhow, so let me go back to how we're expanding the methodology and go from a net compound that I just described to you to this type of compounds that are that we think of them as curved catalysts, right? So essentially here we have changed quite a few things, right? But the point is we're starting from a racemine that then drives the assembly into a metal catalyst, or well, let's not tell it catalyst, a metal complex that has the ligand framework around it in a curved fashion. This is relevant because some of the most active catalysts, specifically for oxygen evolution reaction, is proposed to be curved systems in materials, right? Now, doing this in a homogeneous fashion is extremely difficult, and this is probably one of the few examples where you can do that. So we're still exploring this. We don't have a crystal structure to show you that we have made it. We have MALDI evidence, we have NMR evidence, but we would like the crystal structure, right? I love crystal structures if you haven't noticed it. So I push my students to get crystal structures if at all possible. Anyhow, another type of compound that we've been exploring are these extended nets, right? So we're calling them a super net. And initially I thought this was impossible, but my student Said proved me wrong when he showed me the NMR and later on the crystal structure, which is fantastic, right? This is a compound that is more than 3 nanometers in diameter, right? And again, right, I always ask myself, and I always tell my students, you should always ask, why do we care? Because people are going to ask you, and so you should be the first one to ask yourself that. And so why do we care? Well, we care because a few years ago, there was a science paper in 2019 by the Sobe Lab in Japan, and they reported these phenine nanotubes, right? And these phenine nanotubes were proposed back then, and they have been explored as potential systems where you can modulate conducting and semiconducting properties by essentially controlling these void spaces. And so we're right now hypothesizing if we can still obtain similar properties if we were to do this in a 2D fashion, like using this type of materials, right? Not 3D, but 2D. Anyhow, so let me change gears and tell you a little bit about the other 2 topics that we're doing research in my lab. So hopefully the other 2 topics are a little bit faster. And these are inspired, the next one is inspired in mimicking supramolecular architectures in nature. So how do we modulate metal-metal bonds? Here you have two enzymes, N2O reductase and nitrogenase, both of which are critical enzymes. One of them essentially reduces N2O to dinitrogen. The other one takes dinitrogen and reduces it further to ammonia. Right. So the idea is, in both of these cases, you have close metal-metal contacts, right, in the order of 2.6, 2.5, or you can think about it differently, in the order of the distance between the sum of the van der Waals radii, right, for that given metal. And so that provides to the enzyme cooperativity for the metal atoms to act in concert. So in the lab, what we decided to do is to make a species that looked very much like this cartoon, where we can synthesize a given macrocyclic base, append a given ligand arm, and hope for the nucleation of clusters with this particular topology. So square planar topology to mimic the active site or subsite in nitrogenase. And so these are some of the known compounds with that particular topology. As you can see in all 3 cases, you can argue that there's in the ligand, from the ligand sense, there's a lack of tunability from the ligand perspective. It's hard to envision how to change the ligand nature. You can also argue that the coordination sites are essentially filled, or there's no coordination sites because we have a D10 species in this case, or the ligand is too flexible, so that when subjected to stress, oxidative or reductive stress, likely it's going to fall apart. So what we ended up doing is synthesizing compounds or ligands that have a much more rigid structure that only presents the ligand donors into one particular region in space. And so by doing that, we were able to synthesize cluster with following different conditions as shown there to make this type of copper clusters. So these copper clusters, we reported about 2 years ago. The copper to copper distances are fairly short, in the order of 2.7 angstroms. They actually form a dimer in the solid state, as shown there. And very interestingly, when synthesizing via a similar protocol, the silver 4 and the gold 4, we get an effect where the metal plane to metal plane, as we go down the group, shrinks. And so we're trying to understand what's happening in between the two planes of metal atoms, essentially trying to determine what kind of bonding, if there is any, that is present on top of that, or in between those two metal planes. Because ultimately, we would like to use that orbital density to activate a small molecule. Okay, so now what else did we learn about these molecules? Well, we learned that we can prevent dimerization if we change this particular R group. So it's hard to see in the structure, but these are isobutyl groups appended to the ligand. If we change that to phenyl, then we have a compound that does not dimerize in the solid state. Because now the group is fairly large, and so dimerization does not take place. We know that it has an oxidative event, just about 200 millivolts towards oxidative potentials. We can isolate that particular oxidized species and then start probing its EPR. And then in the EPR, what we see is what seems to be a delocalization effect. So at 80 Kelvin, what we see is a single classical copper 2 site. Now, as you go up in temperature, you start that isolated copper 2 site starts disappearing to give rise to a multi-line pattern, right? So presumably, if you had localized oxidation into one copper center, as you go up in temperature, then that hole is now delocalized across the whole cluster. Now, we don't believe it's delocalized across all 4 copper atoms, but it's likely delocalized across 2 copper atoms and a bridging anyl item. And that is relevant because the electronic structure that we're seeing here is similar to that of copper C in N2O reductase. And this is, as comparing to studies by the Solomon lab, where they initially proposed it was delocalize the whole across 3 metal centers and then later on across 2 metal centers. Shifting to a slightly different ligand, and here the thing that we've changed is essentially just the size. We've introduced that linker and made this cavity slightly larger. And so what happens is, by doing that particular change, we're able to host simple molecules like acetylnitro, something that maybe is not is not that interesting, but by hosting acetylnitro, we know which resonances, and specifically this one's pointing towards the acetylnitro molecule shift. And so if we test a different substrate and a more interesting substrate, in this case dioxygen, we see the shifting of those same resonances, A, B, and C. And so we see the reversible uptake of dioxygen. DFT tells us that it likely sits right there. And most importantly, we can corroborate this with Raman spectra, where the O2 unlabeled, or O16, shows up at about 1584, and whenever it's when we enrich it to 18, we see a shift towards 1556. Not quite the shift that you would expect, however, is the shift in the direction that you would predict. So interestingly, we don't see electron transfer to dioxygen, right? Which is one of the first things that we were expecting, given that this is an all copper 1 species, right? So why is that happening? We don't know, but we may have a hint as to what is involved in that. So keep in mind that in all of these clusters that I just showed you, the metal plane is almost perfectly planar. The distance, the angles between nitrogen, copper and nitrogen are in the order of 177, 178. Now, changing that to a slightly different ligand scaffolds, and so this is a system that we developed a couple of years ago, and we're now working towards the first publication. So it's really just a two-step synthesis, starting from a very well-known ligand, which is cyclin. And so from there, we can synthesize these octaamine species that we use in a similar condition to make a cluster, to make that copper 4. In addition, we can do the stepwise, the protonation, metallation, and we can also obtain the metal 2 precursors. So these are metal 2's compounds, which are relevant in what I'm going to tell you in just a moment. However, that's the crystal structure of the copper 4. Now, one thing to notice here is 2 of the angles are in the order of 172, and the other 2 are at the order of 156. So there's this bend in the copper 4 plane. Now, that is enough to make this species now very reactive towards dioxygen, right? So in the previous clusters, planar, no reactivity towards O2. In this case, you distort the cluster just a fraction, and now all of a sudden is extremely reactive. Now, I would like to tell you the more about it, but this is just right now the qualitative observation, right? Of what is happening. We're working towards seeing whether we can isolate the intermediates, etc. How can we potentially do catalytic chemistry with O2? But for the time being, on this front, I can tell you that we're on track to making hypothetically an iron 4. Well, this is now from very, very recent results. This can be an iron 4 or iron 6 actually. As you can see, in the lab of an assistant professor, things evolve pretty rapidly. Anyhow, so the metal 2s, we're using them because we've seen initial results in that we can still deprotonate those remaining NHs and further, expand the metal cluster. And again, a lot of that is still in the preliminary stage. So with that, I'm going to shift towards the last topic that I wanted to tell you, and that is about anion recognition. I told you at the beginning, why do we care about anion recognition? Well, we care in my group because we would like to get rid of these particular anions. So these are PFAS. PFAS can be anionic or neutral or sweet or ionic. The anionic ones are the ones with the highest solubility in water. And usually they contain one of these 2 anionic head groups, either a sulfonate or a carboxylate. And then it has a polyfluorinated chain that can take a lot of different forms, as you can see here from this database of the EPA. Now we care because if you were to ingest PFAS, it is, as you can see, very detrimental to your body, right? It can cause various types of cancers. It is very, it has developmental effects in the fetus. It has a residency time in your body of anywhere from 4 to 8 years, which is pretty long, right? And some figures, according to the Environmental Working Group, says that 99% of adults in the US have some measurable amounts of PFAS in their blood. Now, how do we even potentially ingest this? Well, if we track them back in time, you can essentially track them to the discovery of Teflon, right? Then after Teflon, for a number of years, it was in use, but then it was figured out that it could be used in these AFFF system, which is essentially that foamy water that you see, or you used to see when firefighters used to train to put out heavy fires, right? So these foams are made with about 2% of PFAS in the water, and you make this type of foam material in addition to a few other things. The point is that from there, PFAS were, their thermal properties, their anti-corrosion, anti-stain, and all of the anti-things that they have, were explored in the making of a lot of different type of materials, right? From cookware, clothing, medical devices, coatings, paints, etc., etc. Right? Now, in the early 2000s, it was discontinued. Its manufacturing in the U.S., not in other places in the planet, but because they were manufactured for about 40 plus years, then they are known to be or to exist in the environment essentially everywhere. And I'm going to show you a data to that effect. Here you have I forgot to put the reference here, but here you have just common items that you find in the supermarket and the levels of PFAS in that particular food item, right? So for example, I tell people, look, if you like iced chocolate cake, maybe it's not a very good idea that you keep eating that, because it has, north of 17,000 ppt levels, right? Now, remember that number, because there you go. Because if we look at the map of the US, and this is a map put forward by the Environmental Working Group, they claim that roughly 200 million Americans have it in their tap water. On top of that, the EPA has established the maximum contamination limit of 4 parts per trillion, right? Just seconds ago, I told you, iced chocolate cake, 17,000, right? So we're clearly at a very different scale here. But this is the recommended value or number that we should be under. So essentially, all of these light blue, places have exceeded that value, that number. And last in this slide, keep in mind this date. This is September 3rd, 2024. This is the date when the EPA is finalizing the new water drinking standards, right? So after that, utility companies, industries are going to have to adhere. They're going to have some time, but they're going to have to adhere to those water drinking standards. And one of the standards is this, right? So an MCL of 4 ppt for PFOA, PFOS. Cool. Now, so what is the challenge that we're facing here? Here you have a chart of common sorbent materials, and then PFAS concentration at a very, very large concentration scale, right? It varies by many orders of magnitude. The problem is here. This is the environmentally relevant concentration. This is also the concentration found in the drinking water that we care about. So 1 microgram per liter or 1 parts per billion. And if you recall the number I told you, right, 4 ppt, that's right here. And so essentially right now, there's no material, commercially speaking, that can take, that can, address this challenge. Here I'm showing you 3 of the 3 popular groups in the US working on this problem. And as you can see from their results, the concentrations, the residual concentrations that they are able to get are in the order of 10 to 20 nanograms per liter. So that's 10 to 20 ppt, right? We're still need to go a little bit further down. And so when in our studies of anion binding, we synthesize a particular host for anions. And I'm not going to go into much detail about the synthesis, but I'm only going to tell you that these particular species are capable of essentially binding an anion right here in the middle of the scaffold. And we tested methyl sulfonate as a kind of surrogate for a PFAS, right? I mean, keep in mind that this is not fluorinated, this is just a methyl, right? But we have the anionic head group that we wanted, so a sulfonate. And what we noticed via NMR is that binding of methyl sulfonate, similar to the data I showed you before, here you have free host that disappears as you titrate methyl sulfonate. And so it disappears to give rise to the host gas complex. Now, that host gas complex is so stable that even if you boil it in DMSO, and this is an NMR collected in situ, you don't see the methyl sulfonate coming off from that particular host, right? So the host gas complex is more stable than the solvation energy that DMSO can provide to the anion at 100 degrees C. So this was the result that pushed us to the next step. That was, well, let's actually test it towards PFAS, right? Those are the crystal structure of confirming the binding and whatnot. So what we did was we had synthesized the system with an alkyl chain, but we needed to turn this into a material. So what we did was a slight modification. My student Swati did a modification where we appended a terminal epoxide into the R group, right? And so we use that epoxide to then polymerize it or co-polymerize it with other species. In this case, we decided to use one of the resorcinerines that we had in hand to make this type of membranes that we're just calling them for short PFMs. And so the idea is that in these membranes, you subject them to contaminated water, and then you start sequestering PFAS in these particular binding pockets. That's the whole idea. And so we tested this, and this was one of the first results. You can see here, we're using PFOA starting at 1 parts per billion, and the residual concentration in that water is going rapidly down in matter of hours, and then it somewhat stabilizes, past 10 hours at levels that are relevant to the problem at stake. So, for example, here at 48 hours, this is well, somewhere here, right? So this is 1, 2, 3, 4, 5. So this is 4, 5 ppt. So we're somewhat getting to the target value, right? Now we also studied other PFAS, other longer, slightly longer, all the way to the shorter one, the C4, which C4 are the hardest to remove at this point in time. So this is the hardest because they have I guess more higher solubility in water. And these are also very important, the 8 carbon long species, because these are the most abundant. So we have 2 problems here, right? The most abundant that we need to get rid of, but also the newer ones that are just simply harder to get rid of. Anyhow, so we tested, well, how many times can we reduce our polymer? And this is essentially just taking the same polymer, subjecting or bringing the concentration of PFOA back to 1 ppb, right? So essentially after each cycle, you go back and then we remove, and then we go back and then we remove. And so the data tells us that we can use it, about 9 times. After then, we go above the advisory limit set by the EPA of 70 ppt. Now, but really, we want to use this in a real application. We would like to then start doing column mode experiments. And so here I'm showing you 2 of the variations of polymers that we have. So these are just PFM1, labeled PFM1, PFM2. Let's just not focus on PFM1 and only on PFM2. And so if we focus on this particular performance and extrapolate, we calculate that with half a kilo of this material, we can clean, and half a kilo, that's about a pound, right? We can clean about 5,000 liters of water. Or said in a different way, this is about the lifetime of a filter for a year, right? If you go to Lowe's, to Costco, to Home Depot, and you get one of those filters, right, to get under your sink, they're usually rated for 6 months to a year, right? And so we're now getting to that level of performance. Hopefully we can extend this further, and that's what we're working on. And the very last piece of information I'm going to give you is that we're now moving into really testing drinking water, right? So you can always think about, well, if we're going to use this, we need to test it in the actual water that we're drinking, right? So we moved to this building just last year, and the filters in the water fountains are brand new, right? Or as new as they get. And we got some of that water just recently and spiked it with PFOA, and then tested our membranes for removal, right? And so this is the residual concentration in the solid circles. As you can imagine, in a real water sample, you have salts, you have some leftover organic matter, right? So there's inherent competition that you don't have when you're using milky water, but that's fine, right? Because this is really the real problem at stake, right? So if we start understanding how to make it better, then this is now, or could be, widely applicable. And so with that, I'm gonna stop. And I would like to thank again all of my students and collaborators, the funding agencies that have made all of this work possible. Those of you listening, thank you for the invitation to speak at GHS, and I'll be happy to take any questions.
I will bring this session to a close. I really appreciate, and we as the Greater Houston section and our society really appreciate you taking the time to speak with us today, Raúl. And I know we all really enjoyed your talk. Hopefully, as we mentioned, for those who are online, we do have more events upcoming, so do check out our website at acs-ghs.wildapricot.org. And we hope to see you again at some future events. And thank you again, Raúl.
Thank you very much. It was a pleasure.